Okay. Welcome those of you joining us for the Centers of Excellence in Hematology Microscopy Monday. We're excited to have you join us, those who've been here before and new individuals. This is a great opportunity to learn about different cores at the Centers of Excellence in Hematology, and also learn methodologies and skills for doing excellent microscopy. And this is also an opportunity for you to pick the brains of those core directors if you have a project that's a microscopy project. So I hope that this is educational opportunity, and we want to really welcome Dr. Bewersdorf for speaking for us again. Before we go to his talk, I just want to share my screen for 1 minute and remind you of opportunities for pilot and feasibility. So this is the Centers of Excellence in Hematology. The homepage, it's cceh.io. And there's lots of things you can go look at here, but I'm going to draw your attention right here to this funding section. And if you go to funding, and this always pops on, so I have to stop that. And if you just scroll down this page, you'll find information about funding opportunities. And we have a new call out for type B applications. There's two types. There's type A and type B. Type A, you can apply for anytime. Therefore, core credits, essentially up to $12,000 worth of money that you can bank at a core, like an imaging core, if you have a project that involves that core. And those can be applied for anytime. We have a second call out for type Bs right now, which are up to $75,000, and they're due February 28th. These are novel directions for investigators. It can involve, it can be a grad student, it can be a postdoc, it can be a new investigator, it can be an investigator who's established, who's going in a different direction. And these are kind of a great opportunity for you to get some funds to help support a new direction or your first direction that you might be doing in a lab. So I highly recommend you go take a look on our website. And I'm going to stop sharing right now. And then I'm going to turn the time over to Dr. Bewersdorf for our talk today on super resolution microscopy.
Thanks, Diane. So yeah, I'll let me share my screen. Here, this one. And then start the presentation. Let's swap my displays. Okay, oops. Okay, so today I want to talk about super resolution microscopy, and I want to give you an overview of that field and talk a little bit about the pros and cons of these methods when it comes to biomedical applications. This is the core expertise of my lab. And something we actively do research on. And towards the end of the presentation in particular, I'll show you examples of techniques that we are developing in this lab. The talk is about, well, probably nearly an hour long. So if there are any urgent questions or so which really stop you from understanding or following me, please don't hesitate to interrupt me. But I'm also around for kind of the next hour after the talk if there are any questions you might have. So we have plenty of time to discuss this if you have the time after the talk. So the goal, yeah, let me just jump right into it then. So we talked a few months ago already about like the basics of microscopy and of confocal microscopy, and the concept I introduced then was that of diffraction and of the point spread function, and that is really the motivation why we are even talking about something like super resolution microscopy today. So the problem we deal with in a microscopy, and from a physics perspective, we mean far field microscopy, which means lens-based microscopy, where the lens is a certain distance away from the sample. This is not scanning probe microscopy where you have some kind of tip which you move across the sample. So in these lens-based microscopes, we we suffer from the phenomenon of diffraction. And that means that even the smallest possible object we could image, let's say a single GFP molecule or so, by the time we detect it, will appear as a blurry spot on our detector. So we call that the point spread function, which describes how a single point appears in our image. And a typical pattern that you could observe is, for example, this airy pattern here. So, and this diffraction limit really is a fundamental physical limit, which already was described 150 years ago by Ernst Abbe and others, where he observed from a mathematical perspective that you cannot resolve structures smaller than about half the wavelength of light with a microscope. And if you want to put this in a formula, you can, for example, describe here the radius of this area disk, like the center to a first minimum here, distance here, and that is 0.61 times the wavelength divided by this numerical aperture. We talked about the numerical aperture a few months ago. I encourage you to go back to the recording if you want to refresh that a little bit. But this is essentially of the order of about 1. The refractive index is somewhere between 1 and 1.5. In this aperture, this sign of an aperture angle is, well, never bigger than 1. So numerical aperture, think of this for our discussion today as about 1, and then you have about half the wavelength here. So how does this diffraction limit or diffraction limited resolution now compare to what we are actually interested in when we use microscopes in biological imaging? And here the subject of choice is typically the cell. And the diameter of a single cell is of the order of about 10 micrometers, sometimes tens of micrometers, but this is a typical dimension. And the point spread function, so the smallest resolvable spot, is about this size. It's about 250 nanometers across, so a quarter of a micrometer, and about 500 to 700 nanometers or so in the depth direction for these high numerical aperture objectives. And that's about the dimension of, or the diameter of a mitochondrion. So you can really resolve the individual organelles in a cell and separate the nucleus from mitochondria, etc., without any problems in a conventional light microscope. But when it comes to going down to the molecular detail now, for example, we zoom into here the plasma membrane and we see here individual receptors and channels here. Then we are talking about a 10 nanometer and not a 10 micrometer scale bar. And this point spread function is really far out of proportion over what we would like to see. So this is where then super resolution microscopy comes in. This is summarizing a large variety and range of techniques that all attempt at improving this resolution so that we get closer to seeing these molecular details inside a sample. So I'll start with an overview of the different techniques that exist and put them in context to the resolution we can get with them. And then we are diving into the details of some of these techniques here. So here I'll plot the example of a mitochondrion and plot the point spread function of a diffraction limited standard microscope next to it. Okay, so as you can see, this is about the diameter here of a mitochondrion and 250 nanometers in the focal plane, the XY direction, and about 600 nanometers in depth. We call it the Z direction. And that already assumes that we have a high numerical aperture objective. We have a range of techniques which really push the diffraction limit to its limits here, I would say. Where we squeeze out a little bit more information out of the optics of the microscope or the image processing to really get to a little bit more detail here. And first, we have deconvolution techniques, which is mathematical post-processing of data. And there we get about a square root of 2 resolution improvement typically. And then we have the structured illumination microscope, which projects a stripe pattern into the sample and gets about a 2-fold resolution improvement. And then we have more recently developed techniques like the area scan microscope and the ISIM microscope, which range in resolution somewhere between the deconvolution and the SIM microscope, and they can also be combined with deconvolution to get closer to the SIM resolution level. We then have the groups of techniques which really break the diffraction limit here, and which were awarded the Nobel Prize in 2014 in chemistry. And there we have the STAT microscope as well as the single molecule localization techniques. On the STAT microscope, the resolution discussion is a little bit more complicated because you can either optimize your XY resolution to the maximum. You get about 10 times better than the diffraction limit, but without any improvement in Z direction. Or you optimize the resolution in 3D, and then you get a more isotropic resolution of the 100 nanometer scale. In the single molecule localization techniques, we get even better resolution, but it comes, as you'll see later, at the cost of being fairly slow on the imaging side. And we get resolutions here of the 10 to 50 nanometer or 50 to 100 nanometer scale. And if we use two opposing objective lenses, we can get it down to an isotropic resolution at 10 to 20 nanometer range. You might know these techniques under the acronyms PALM, STORM, DSTORM, FPALM, or lately DNA paint, and I'll talk more about that later. And then there's an exciting new technique around for, I don't know, 5-7 years or so now, called MinFlux, and that really gets you down into the single-digit nanometer range. I won't have time to talk about that today, and the reason why I excluded this, it's fairly complicated to explain, but also the system availability in the United States is still fairly limited, and we don't have access to one of these systems here. And the last group of techniques I'll cover today is that of expansion microscopy. And expansion microscopy is very different from all the other techniques, namely that it's not an optical technique to improve the resolution, but a sample preparation technique. You're physically inflating your sample, if you like, and that leads to a respective improvement in the resolution, which is somewhere between 4-fold to 20-fold resolution improvement. And we'll conclude today discussing that. Okay, so I'll discuss deconvolution, Aries scan, and SIM microscopy briefly. I'll spend a little bit more time on STAT and then the single molecule localization techniques, and then I'll conclude talking about expansion microscopy. So first, deconvolution microscopy. The idea of deconvolution microscopy is to amplify sharp features that you have in an image by image processing. And this is mathematically gets quite involved, and I don't have time today to really cover this. Important to note is it's an image processing method. And I just want to show you a couple of examples pointing out that there's really a myriad of deconvolution algorithms around, each having their own strengths and weaknesses. And here you see a few examples taken from this website here. So they all result in some resolution improvement and also in some background reduction. And these are two of the advantages that we are often like experiencing in deconvolution microscopy. And just to summarize here the deconvolution microscopy, it is the advantage of deconvolution microscopy over all the other methods we are talking about today is it's post-acquisition. So you can combine it with any microscopy method, microscopy technique, and you can even do it after the fact, right? If you later discover, oh, it would have been nice to get a little bit more resolution out of my data. It's also easily accessible. There are commercial packages which are very powerful, but even programs like Fiji or ImageJ have fairly advanced deconvolution packages included. And one of the nice side effects of deconvolution, and many people use it just for that purpose, is that it's also very good to remove any kind of out of focus background or blur that you might have in your images. The disadvantages of deconvolution microscopy from a user perspective is that the resolution improvement is moderate. It's, I would say, typically about a square root of 2. You can push it maybe to a factor of 2. So square root of 2, you can push it to 2. But it's prone to artifacts. So especially the more you push the resolution improvement. So you can, at some point, if you push the resolution improvement here too far, you can create structures out of just noise. So these are not real structures, it's really just artifacts. But it's very hard to distinguish these artifacts from just random background. So, and the noisier your data is, the more difficult it is to identify these artifacts, and the more prominent they will appear in your image. And that makes the deconvolution microscopy somewhat problematic at a quantitative level, since you can't necessarily rely on interpreting every detail accurately. Some of the algorithms are not retaining relative brightnesses, so an object which was twice as bright as another one after deconvolution might not be twice as bright anymore. It might have changed different brightness level. So it's difficult to analyze or quantify signal levels, for example, after deconvolution. And structure sizes, diameters of structures can also change. Deconvolution tends to make everything sharper, whether it was intended to be sharper or not, it doesn't matter. So you need to be somewhat careful with the interpretation there. It is very useful to enhance images for qualitative presentations, so to just highlight what you want to show in a particular data set. It's good to inspire new hypotheses you might have, because it makes you see things which otherwise would have been hidden. But you should follow up with some other alternative methods to really confirm that what you have seen in the deconvolution data is really it's real. So use it with caution when you draw any quantitative conclusions from deconvolved images. So with that, I want to move on to the next topic here, and that's area scan microscopy, which is very popular, very prominent now. Most imaging microscopy cores now have an area scan confocal microscope from size. And the idea here is you get the resolution of the perfect confocal microscope, which normally would require you to close the pinhole and here, and then you lose a lot of light. Here you don't need to do that, and you can get the resolution of the perfect confocal without losing that light. So let me briefly remind you how the confocal microscope works, and then based on that I can explain how the area scan microscope works. So this was already included in my presentation a few months ago. So in the confocal laser scanning microscope, you have a laser beam which you focus into your sample, and there it creates a laser focus, which is diffraction limited. So you get this kind of distribution of light intensity here. And this is the optic axis is now in this direction, similar to how it's shown in this plot. And then you detect fluorescence here being emitted from the focal plane through a pinhole here. And only if the light is emitted here exactly from the focal center, it will go right through the pinhole. Light that is emitted from other planes is mostly blocked by the pinhole, and only small fractions of that go through the pinhole. So the pinhole now has a detection efficiency depending on where the light originated from, and it looks very similar to an excitation focus. So now you combine your excitation probability given by the laser intensity with the probability of detecting light from a certain area on the focus, and you get the overall points of function of your system. The overall probability of detecting light in a sample from a certain position for a certain laser focus position. So as you can see here, the laser scanning microscope is very good at filtering out this out of focus light, and that's what we emphasized a few months ago in my presentation. However, it also improves the lateral resolution somewhat. If you look at these two images, right, the edges here, which are already dim, will become even dimmer when you multiply it with this function, since that's dimmer here than it's in the center. So you get a little bit of a resolution improvement in the perfect confocal microscope. And then you can also see that in this representation. Same thing here, just now the points and functions are rotated by 90 degrees. The optic axis is in the z direction here now. So here that's the excitation focus. This is the detection Poinsett function. If you multiply the two, you end up with a slightly sharper Poinsett function. So what this representation neglects, however, is that you have a pinhole. So if the pinhole is very small, nothing really happens. So we essentially need to blur the emission Poinsett function here with the size of the pinhole by doing a so-called convolution. And as you can see, it essentially looks the same if the pinhole is more or less like a point. However, if your pinhole is open to about what we call 1 area unit, then, so it's about the size of this distribution here, then you get a significant broadening of this detection Poinsett function. And if you now multiply this one with the excitation Poinsett function, you'll see that we can still suppress the background light up here and down there, but the resolution improvement is really non-existent anymore. This is as sharp as this one here, right? So the reason why we open the pinhole is so that we can actually detect any significant amount of light. If we close the pinhole, then only a few % of our fluorescence will pass through the pinhole, and the images will get so dim that you cannot essentially work with a microscope. So using a pinhole size of about 1 area disk size is what we typically use. So we now have this dilemma that we need a very small pinhole to get that resolution improvement, but without an open pinhole, we don't see anything. So the solution that the area scan microscope introduced was that you use multiple very small pinholes, and then you detect an image with each of these individual pinholes here, and then you generate the image by combining all these individual 30 or so images into one new image. So here, this is the detector that's used in these size microscope, area scan microscopes. As you can see, each pinhole is about 0.2 area units here, like even smaller than this one, while the overall area that is covered is more than 1 area unit, so more than 1 diameter of this Poinsett function. So there's, however, one problem that we still need to solve. We get 32 individual images, but each image will show something slightly different because they are detecting light at different positions. So, and there, fortunately, we can take advantage of one very simple fact, namely that if the pinhole is shifted by a certain distance x, the image that you get from the laser focus at the original center position and multiplied with that shifted detection Poinsett function is just shifted by half that distance, and it has the same shape. That function has the same shape as the non-shifted one. So here the green line is the effective Poinsett function that you get if there's no shift between the pinhole and the laser focus. Here the green function is what you get for a shifted detection Poinsett function. Here the red line where the laser focus still stayed at the original position. And as you can see, it shifted by half the distance, and the shape looks the same as this shape. So what you can now do is you take all these green images. You know their shift because you know the detector, you know the dimensions of your detector. And then you take all these individual green images here and you shift them back to the correct position and you add them all up. And you do that computationally. That's all done in the background by the microscope. If you do that, you get this 1.4-fold resolution improvement that you essentially get if you multiply two functions which have the same shape. So the square root of 2 resolution improvement. And you can also combine with the deconvolution, and then you get typically a 2-fold resolution improvement. So here's just a proof of concept image here from that size white paper here. This is the regular confocal image. And here you see how just simple Wiener deconvolution algorithms would perform. You definitely see more details, right? This is here. You see already a little bit of a ring here. It becomes more prominent here. But with the area scan on top of it, you get even more resolution improvement. Now you see that all of these are ring-like objects. So, so in summary, just a few words about the history here. So the idea is old. It was first invented by a kind of very prominent microscopy theorist, Colin Shepard, in 1988. He published it in a kind of a optics paper. Look, isn't that a curious little thing here, that the Poincare function is shifted if you shift the pinhole? And then it was rediscovered about 10, 15 years ago. Actually was discovered multiple times. And we have this image scanning microscope, these ISIM, the rescan, and the OPRA systems. They all are based on this technology, and as is the size area scan. So the advantage of these techniques is that it works with any regular confocal microscope sample, as does the deconvolution. And you get this resolution improvement without compromises in signal, so you don't need to close the pinhole here. And while the size area scan has to do some image processing somewhere in the background, some of these techniques like the ISIM, the rescan, and the OPRA technically don't even need any image processing. You already get that resolution improvement directly. The ISIM is, and the rescan are also commercially available systems. But it's fair to say that the size area scan is much more abundantly available, probably. So the disadvantage of the area scan technology is that the resolution improvement is somewhat moderate. You get kind of this about square root of 2 effect. If you combine it with deconvolution, you get about a factor of 2. But if you combine with deconvolution, which is the default setting, unfortunately, you also, of course, need to worry about deconvolution artifacts again, since it's kind of built into the process. So again, be a little bit cautious with the interpretation or over-interpretation of these area scan images if what you are looking for is really at the diffraction limit or below. Okay, so then let me switch to the third technique, and that is a structured emulation microscopy. This is a little bit older than the area scan, but somewhat related here. So what we do here is we illuminate our sample with a pattern of light, and then that allows us to separate neighboring objects more easily. So let me give you a kind of a very rough overview of how we do this. So let's say this is the structure here that we want to image. It consists of these two, let's say, microtubules here. And we illuminate it uniformly in just a lamp-based wide field microscope. So then all these microtubules get excited at the same time. They all emit light. Each point of this structure will become blurred on the way to the camera. And you see a blurry pattern of these two microtubules. And as you can see, when they get too close together, you cannot separate them anymore. So what happens now if you illuminate your sample with a striped light pattern? So now what happens is that the stripe, once here, one of these microtubules happens to be in the minimum between the two stripes and is hardly excited, but the other one still is excited because it's overlapped with this area here. So again, if we blur this on the detection, we get a blurry image, but only one of the microtubules essentially shows up. And now we can move that stripe pattern, and you see how we illuminate different parts of the sample with the stripes, and that is also represented on our recorded image. Now what we can now do is we can essentially add and subtract these three images from each other, and we get an improved resolution image by taking advantage of the different information that these three images have. And what I'm not showing here, because this is vastly simplified, you're normally also rotating your stripe patterns by 60 degrees and 120 degrees so that you get that same resolution improvement effect also in the other dimensions. So you typically take somewhere between 9 to 15, in the newest versions, 8 images that to get a super resolution image. This is overly simplified, but I think it gives you a little bit of an idea of the concept of the structured illumination microscope. Let me spend 1 minute to go a little bit more in depth here. And what we are taking advantage here of is a phenomenon which we know in digital life nowadays as moire patterns. So if you ever watched kind of a moderator on TV wearing a striped shirt, and you have an old-fashioned TV set with not that many pixels, then the stripes would often appear, and you would see these very weird kind of oscillating stripe patterns on the shirt when the shirt was moving slightly. What you essentially have here is that you have one stripe pattern overlaid with another stripe pattern. And what happens mathematically, if you multiply one stripe pattern, like one cosine function with another cosine function, is you can replace that by this trigonometric identity, which results in the difference between the two frequencies and the sum of the two frequencies. And the difference, so high resolution means high spatial frequencies, or high level of details means high spatial frequencies, many stripes, right? You cannot observe them, but you can observe the difference between the two frequencies here. As you can see here nicely, right? These wide black stripes, that's this difference frequency, can be easily seen, where the others become quite hard to see if your resolution isn't that good. So this is, you can plot this more generally in Fourier space, and I don't have time to go into that, but encourage you to look here, for example, at this link down here, if you're interested to learn more about structured illumination microscopy. Instead of going into the details of the theory, let me just show you one example, and then let's talk about the pros and cons. So here's a cell nucleus where the DNA was labeled with Darby or Höchst. On the left column, you see regular wide field microscopy. This is the top down view. This is a side view. And then here you see the deconvolved data set. You already see more details. And then here in the structured illumination data set, we get about a twofold resolution improvement. You now nicely see here, when you focus on the face of the nucleus, you see the channels of chromatin behind the individual nuclear pores. So there you have kind of a gap in the chromatin, so that I guess the RNA and other molecules can more easily pass through. So this becomes nicely resolvable here. In the side view, however, you already see one of the challenges with structured illumination microscopy, and that is you are prone to these artifacts, especially for thicker three-dimensional objects, right? You see these ghosts emerging here at the top. You also see it in the deconvolution data actually even more prominently. And this is something you always need to watch out a little bit when you do structured illumination microscopy. However, it is a very great technique here, and the resolution improvement is impressive, and you can easily combine it with multicolor imaging. So here's, for example, a three-color imaging where people looked at the DAPI, the lamin, and nuclear pore complexes here. So the advantages of structured illumination microscopy is, especially compared to the later techniques I'll talk about, is that it works with all popular fluorescent labels. So it's a good choice especially for multicolor super resolution imaging. I just showed you 3-color images in the previous slide. And it essentially works with your standard sample preparation protocols. It's also relatively fast. You need to take 9 or 8 to 15 images, but that's only 8 to 15 times slower than the normal wide field microscope. So it's still fairly fast, and people have successfully used it for live cell imaging as well. And it works quite well for 3D imaging, as long as the sample is not too thick and too bulky in its staining. The disadvantage is that the resolution improvement compared to the methods I'll talk about next is only twofold. However, this is the fact in all three dimensions. So the volume resolution is 8-fold better, 2 times in X, Y, and Z. And it requires image processing to make sense of these 8 to 15 striped images that you recorded. And again, that means it's prone to artifacts, and you need to watch out before you take subtle details too seriously in your interpretation. And it also doesn't work well with high background situations, and that's particularly a problem if you have thicker samples, in particular when you work with tissue. But for cultured cells or for very sparsely labeled 3-dimensional samples, structural mediation microscopy works really well. Let me change pace a little bit and talk about now how to really break the diffraction. What I've talked about so far are all methods which push the diffraction limit to its real limits. Okay, you squeeze out a little bit more information to get the square root of 2 or a factor of 2 resolution improvement. But you can really break the diffraction limit and get 10-fold resolution improvement. And the secret here is that if you cannot separate 2 molecule emissions in space because they're too close together, you can separate them in time and image one after the other. And to do that, you switch molecules on and off so that they are not emitting light at the same time. So this was here subject of the Nobel Prize in Chemistry in 2014, which was exactly on this topic. And the first technique I want to cover here is STAT microscopy. And then the second group of techniques are the single molecule localization microscopy techniques. So in the STAT microscope, we break the diffraction limit by switching individual molecules off in a very targeted manner. And STAT stands for stimulated emission depletion, which is the process we use to switch off molecules. So what is stimulated emission depletion or stimulated emission? So this is the energy diagram of a dye molecule. So we excite it by absorbing, for example, blue light. And then you get some vibrational relaxation of these vibrational excitation states. And you get the emission of fluorescence shortly after. And again, you get a little bit of vibrational relaxation down here. So you lost a little bit energy here and here. And the fluorescence emission that you have is therefore a little bit green or red shifted from the blue excitation. So you get green fluorescence emission. So that's the regular fluorescence process. In the STAT microscope, before the fluorescence can occur, so in the first 1 nanosecond, so after we excite the fluorophore, we illuminate the molecule with a second STAT laser, with a second laser, which we call the STAT laser. And this laser can stimulate emission instead of waiting for spontaneous emission of the molecule. So stimulated emission will create an identical copy of the incoming photon here, and will force the molecule down into the ground state without the emission of fluorescence. And this might sound exotic, but it's something which we use daily nowadays, because the S and the E in the term laser stand for stimulated emission. So our laser beams that we create are done exactly that way. You create many, many photons, which all have the same color, all go in the same direction, and that is a laser beam. So in this case, however, we don't care about the stimulated emission light. We care about that we found a way to prevent the emission of fluorescent light. So we turned the molecule off before it could emit fluorescence. So now how do we use this in a microscope setting? So we have a laser scanning microscope with an excitation focus, and that will excite fluorescence across the whole area of the excitation focus. However, before that happens, we overlay our excitation focus with this STAT laser focus, and we make it ring-shaped so that it only overlaps with the excitation focus at the periphery of the excitation focus. And that will now be able to turn off fluorescence in the outer area of the excitation focus and will lead to a sharpening of our the area from which fluorescence can still be emitted. So this by itself is a moderate effect. It's not particularly impressive yet. It's again about a square root of 2 resolution improvement, because the problem is that the donut hole here of that ring is also diffraction limited. So it's still about 250 nanometers in size. However, what we can now do is we can run this depletion process in saturation. We crank up the laser intensity here by making it brighter and brighter and brighter, and that will saturate this whole depletion profile. So if you go to higher and higher intensities, even 25% or 10% of the intensity is still sufficiently bright to essentially turn off all the fluorescence. So we now turn off fluorescence everywhere, except in the very center of that ring, where the intensity ideally is 0. And 0 times the very high amplification of the intensity is still 0, right? So, so now the area from which fluorescence can be emitted becomes smaller and smaller the more we crank up our STED laser intensity. So the way we implement this is we integrate it into a confocal laser scanning microscope. So we have an excitation focus here, and we detect our fluorescence through with a typical detector using a pinhole here. But we couple in an additional laser beam, which is the STED laser, and we put a phase mask into this beam path, which creates this donut shape. So the phase mask is delaying here the light going through from 0 to 2 pi, or from 0 to a full wavelength delay if you go once around here, and that creates this donut shape. Looking at the donut from the side shows that this essentially creates a channel, a dark channel in the middle, and that leads to this strong resolution improvement in the XY direction, but not in the depth direction. So that's why you get this. I showed you in the overview slide in the beginning, this very narrow, long, effective focus here. There are other donuts you can produce that also deplete here the top and the bottom, and that way you get this more isotropic resolution, but it will come at the compromise of a less good resolution in the XY direction. This one is the one which is mostly used if people can work with 2D applications, but the other is a way to get 3D super resolution. But I'm not talking more about that because it's much more rarely used in practice. But I want to spend one more word here, minute on choosing the right dyes and the right wavelength here, because you need to consider here the spectrum fairly carefully when you do STED microscopy. So here you see the absorption spectrum and the emission spectrum of the silicon rhodamine dye, just as one example. And you pick your excitation wavelength close to the peak of the absorption spectrum, as you typically do in fluorescence microscopy. And you detect the fluorescence here in this window, again, fairly typical for a fluorescence microscope. The depletion wavelength, however, you need to pick very carefully. It's about 1,000 times brighter than the excitation wavelength, and you really need to watch out that it's not overlapping at all with the absorption spectrum. If it's even overlapping with just a little bit, then it's already exciting the fluorophores rather than turning them off. So you need to avoid overlap of the depletion wavelength with the absorption spectrum at all costs. So it needs to overlap, however, somewhat with the emission spectrum. Otherwise, you cannot deplete the molecules. And you also need to pick now, even if you found a dye which now fulfills these criteria, you need to make sure that it's also bleaching resistant under these high intensity STED illumination conditions. So we are typically limited by the choice of lasers we have on a STED microscope, which means you need to pick the dyes matching to the lasers you have. And that means you need to use care when you select the fluorescent probes you want to use in a STED microscope. In practice, I would say go with the dyes that have been published by others that work well. Look at images that you liked and say, I want to do the same, then use the same dyes. Okay, let me show you a few examples here. So this is a confocal microscope image of the endoplasmic reticulum and the mitochondria in a cultured cell. We are just looking here at essentially a lamellipodium of a cell, so it's essentially two-dimensional, and it's about like I don't know, 10% of the cell. So this was the confocal image. This is the same area image now in the STEP mode, right? So you get a quite dramatic resolution improvement, and the beauty is you get it directly out of the microscope. There's no image processing involved here, right? So you get that, you just turn on that additional laser, and that's the output that you get. You see now here a little bit of clustering here. This is the individual and the heterogeneity of the antibody staining that we used here that you can now start to resolve. Just one more example here. Here are from Stefan Hell's lab, who is one of the Nobel Prize winners. You see nuclear pore complexes here, and you can resolve the 8-fold symmetry here of the spokes, and the center here in the middle in a different staining. And there's a dramatic resolution improvement here to the confocal image that you would normally get. And you can do it in live cells. This was recorded in our lab. We again look at mitochondria and the ER. And this is playing at, I think, twice the speed. So we recorded one frame every 2 seconds over about 30 frames. As you can see, over time, the image just gets worse and worse because the sample actually bleached over these 30 frames. And we only compensate for it by essentially compensating for the bleaching by just making the image brighter for the later frames. And we also apply a little bit of deconvolution here to make the image look a little bit prettier. But this was not for quantitative analysis, but to essentially show you the dynamics here. Okay, so in summary for STEP microscopy, we can get a resolution which is about 5 to 10 times better than that of the confocal microscope in 2 dimensions. In 3 dimensions, it's about 100 nanometers in 3D, if everything works well. You can work with standard sample preparations, but you want to pick your fluorescent probes carefully. It's relatively fast. I just showed you an example of an image every 2 seconds, and that makes it live cell compatible. However, you need to watch out for bleaching and phototoxicity. It's also a confocal microscope. That means you can work in tissue, and you can because it filters out background light from planes above or below. I mentioned the bleaching as a disadvantage. I also mentioned the fluorescent probe selection. And on the 3D side, it also is not as easy to do than 2D, because now you need to take many, many more images for a 3D stack. And since bleaching is an issue, that makes it quite hard. But it is doable, especially for fixed and well-embedded samples. Okay, so the next topic in this, or next technique in this category here is that of single molecule localization microscopy. You might know it under the acronym PALM or STORM, etc. And then the DNA paint, I'll also talk a little bit about. And here we are not using a targeted switching process like we did with this donut in the STEP microscope, but we rely on stochastic switching of individual molecules. So here's a simulation of what I mean by that. So this is the pattern we want to resolve. There are, I don't know, several 10,000 molecules here. And this would be the diffraction limited image if they all would emit light at the same time. However, if you make them blink so that only a very small subset is on at any time, you can actually see every single molecule here. You find its center position, and you just plot the center position in our sample, in our image. So over time, you will accumulate the super resolution data set that way. The resolution you can obtain is of the order of that of the STEP microscope, 25 to 50, sometimes a little bit better, or with the newer methods, especially a little bit better. And it can also be done in 3D. But it involves quite a large amount of image processing, as you can imagine, right? For each frame that you record, you need to identify and cut out each individual molecule. you then need to fit a model function to this distribution to find the center position, and then you plot the center position in your final image. So if your image consists of 10 million localized molecules, you need to do that 10 million times. So the question now is, how do you make the molecules blink? And the classical way is that you do photophysics here. You essentially either work with photoactivatable fluorescent molecules, like, for example, here in fluorescent protein side, EOS or Maple, or use photoactivatable Geneva fluors, for example. Or you make your dyes photoactivatable by putting them into oxygen scavenging buffers, which allows you to push them into long-lived dark states, and then you rescue them from these states and to make them blink that with time. I don't have time to really go into depth here, but here's a list of good fluorescent probes we are using, and the red ones are here, especially popular. There's another way, however, which is a little bit newer and which is very promising. I want to talk about that in a few slides, and that's the so-called pain probes or DNA pain probes. And here we rely on binding kinetics, so chemical switching, if you like, rather than photophysical switching. And I'll talk about what I mean by that in a minute. But first, let me show you an example of a 3D data set recorded with these photoswitchable or blinking Alexa 647 dye molecules. This is an artistic rendering of the recording process, but you see here the molecules blinking, and then you localize them here in each frame, and you plot their position, and you can image different sections in that same sample and then stitch it all together into this 3D data set. In this case, we are looking at a mouse spermatocyte where we labeled the synaptonemal complex, which has a characteristic structure in meiosis here. You can nicely see now that you can resolve this synaptonemal complex, which consists of these two strands, which are about 150 to 200 nanometers apart. You can easily resolve that here with this microscope, which in this case offered about 20 nanometer 3D resolution. Each dot here, each little cloud here is a single molecule that you localized. I just heard kind of a voice for a second. Is there a question?
I don't see anything in the chat, but if you have a question, please speak up.
Okay, then I'll move on for now. And as I said, I'm more than happy to stay on the call for more questions later. So DNA paint is another technique here. This chemical switching technique I talked about, and I think that's very promising and becomes very popular here. So what you have here is you have a single-stranded DNA with a dye attached to it, and that's your imager probe. And it diffuses so fast in solution that you cannot essentially see it. However, if it binds its complementary docking strand here, which is attached by an antibody to a protein of interest, then it becomes immobile for a short time, and it stands still, essentially, and that's when we can see it and we can localize it. It produces a kind of a flash of light at a certain location in the sample, which represents the position of the molecule. So that's the on state now, and this is the off state, if you like. So the advantage here is you don't need to mess with photophysics. You can pick the brightest fluorophore you can find, optimize the buffers to make it as bright as possible. You don't need to worry about, do they still blink? And if one of these molecules bleaches, you don't care, because it's essentially replaced by another molecule from solution. The only disadvantage of this technique was that that it took very long. You couldn't go too high in concentration here because that would increase the background too much. And if you keep the concentration low, that means you have not that many blinks, and that means the blinking is slow. And it takes hours to acquire a high quality image. We have recently published this year actually a paper where we introduce a modified version of these DNA paint probes, where we put a quencher on the other side of that imager probe. And that keeps the molecule in the off state when it's not bound. And you get a 77-fold improvement in signal when the molecule binds here to the target, because now the quencher is far enough away from the fluorophore that it's unhindered, essentially. This concept might remind you of molecular beacons. So here's a quick summary of the differences. I don't have time to go into that. But in the end, I just want to say this allows us to really be about 25 times faster than with the traditional DNA paint probes. And you don't need optical sectioning like turf microscopy, so you can use it for 3D imaging quite conveniently. And now this gets you into a ballpark where you can also take, in a few minutes, you can take a high-quality image similar to the STORM and PALM methods. But with the added advantages of better resolution and better being resistant to bleaching. So if you're interested in this field, I'll recommend that you check out this paper or talk to me. I'm happy to chat about that. So in summary, the advantages and disadvantages of the single molecule localization microscopy techniques is that of the methods I've introduced so far, this is the one which offers the best resolution. You get down to 20 nanometer and even better with the latest methods. And it's single molecules you're seeing, so you technically can count single molecules. There are caveats attached to it, for example, molecules blink multiple times, so you need to correct for all that. But it is a very powerful and fascinating technology there. The disadvantage is you need to make the molecules blink, and that means you need to carefully select the labels and the buffers. DNA Paint made that a little bit easier, and it's not that fast. It takes 5 to 60 minutes realistically to take a good image of a field of view. And the data processing is also fairly complex, and you need significant training to get good results out of that, which you can trust. So let me finish here with one last topic here, and that's that of expansion microscopy. And that's one of the techniques I'm particularly excited about, but we also need to disclose that I'm involved here in a startup company in this area. I had that on my title slide. So keep in mind that I might be somewhat biased here, but I'm really excited about these techniques. And I hope to convince you that this is really an interesting alternative and very powerful method. So expansion microscopy is a technique which was invented in 2015 by the Boyden lab at MIT. And here the sample is physically expanded 4 to 5 times in a hydrogel. So what you do is you have your biological sample, you embed it in a hydrogel matrix. Okay, you put these monomers in there, you let them form a polymer network around everything, very dense, in a 10 nanometer or so, or smaller size of like the mesh size here. And this obviously is done in a fixed sample, so this is not lifestyle compatible. In these original, so you then expand that hydrogel after you homogenize the sample so that it can expand, and by swelling it, essentially, you wash out all salts, and the hydrogel will just swell and take the sample with it. So initially, the mechanical homogenization was done by using a polymer by digesting essentially the proteins here in the sample by using proteases here. That means after the expansion, there's not much left, which you can image. So to overcome that, you use antibodies before you do that protease step. And these antibodies had DNA handles, which you would link to the hydrogel. And the DNA handles would survive that whole process. You can then later label these DNA handles, and you get a classical fluorescence image. So a year later, there was an alternative process published where you just denature your sample rather than digest it. And that retains the proteins in the gel. And you can also do post-expansion labeling, which has a number of advantages, namely that you can the antibodies are not limited by a crowded environment, and the labels are not expanded anymore. So these protease-free approaches were very important development there. And then there was also the development of iterative expansion, where you first expand the sample 4 to 5 times, you then put a scaffold around it to stabilize it, and then you expand it another 4 to 5 times. And then you get about 20-fold expansion. The initial version was, however, limited to the initial version here with the proteases here, and was not retaining proteome. I'll talk about our method in a minute here, which solves these problems. But let me first show you here a few expansion microscopy images. This is a brain tissue section. And here, this is after expansion, before expansion. One of the challenges you have to overcome is, if you do pre-expansion labeling, is that the fluorescence needs to be preserved. So you either need to choose dyes or fluorescent proteins which survive that whole process, or you need to make compromises in your expansion process so that that you're not destroying the labels, or you use the post-expansion labeling that I mentioned on the last slide. The second challenge that we have in the expansion microscope is that you need to confirm that the sample actually expanded uniformly and isotropically. And you do that by essentially comparing a structure before and after, and see if it's still in the same location in the cell nucleus, for example, here, and that it still has the right shape, right? So there are processes, and this actually has been shown that if the protocol works well, if it's optimized, you can really get structural preservations down to the tens of nanometers of scale very reliably. So the third challenge that we have addressed with our pan-expansion microscopy method that I'll introduce in a second is that these labels are classical fluorescence microscope images, right? So you see what you always saw, namely the protein that you labeled, but not the context of the cellular surrounding, which is a problem that all of fluorescence microscopy has. And the pan-expansion microscopy method, which is a variant of expansion microscopy, really overcomes this problem. And I want to spend the last few minutes, I'm realizing I'm over time, I apologize for that, but I want to talk about that briefly. So in the pan-expansion microscopy is an iterative expansion method, where we expand the sample once and then expand it the second time. And we retain the proteome by using just heat and SDS to denature our proteins, but we don't use any proteases which would destroy the proteins. And since all the proteins are still left in the final expanded sample, we can then label the complete sample with what we call a pan staining. For example, an NHS ester, which labels all primary amines, and therefore labels essentially all proteins. And you get now images which look like correlative light electron microscope images. So how do these pan stainings look like? So this is a NHS ester label applied to a non-expanded sample. And this is just a reminder why we normally don't do this, right? So it essentially shows the whole sample just more or less uniformly lighting up. You see a few like small objects here, but that's not really of interest usually. If you expand your sample, however, twice, and now we zoom in a little bit more here, you actually start to see ultrastructural details here. You see the crystalline mitochondria, you see the Golgi cisterna here lighting up here quite nicely. And this is now a recording just done with a standard confocal microscope, and that's what excites me so much, because everyone can essentially do it in their own lab. You don't need access to a fancy specialized instrument here. So since this is done on a confocal microscope, we can take a 3D stack now fairly easily, just by scanning the sample in 3D. And you can go through here that HeLa cell here. You see it's on the mitochondria, you see the nucleopore complexes, you see the nucleolus here, the Golgi complex. You see all these ultrastructural features inside the cell. And are they, does it make sense what we're looking at? So here we now invert the color table to make it look more like electron microscopy, and we see the centriole lighting up very strongly here in the expansion microscope samples. And it looks remarkably similar to what people have seen in the electron microscope. We don't achieve the same resolution scale, but this is now just done with a normal confocal microscope, not an electron microscope. And we can also look at nuclear pore complexes here, here in the pan expansion microscope sample, here in the electron microscope. But since this is still a confocal fluorescence microscope, we can combine it with an antibody staining. For example, here we are just looking at this polyglutamylated alpha tubulin here. And I can see where it is now located inside the centriole. So hence the comparison to correlative light electron microscopy. So just to show you a few more examples of antibody stainings on top of our pan staining here. Here, mitochondria now, a few examples. TOM20 sits on the outer membrane of mitochondria, as you can nicely see here. This MIC60 sits at the cristae junctions on the inner mitochondrial membrane, right? It's much closer here to the cristae here. And this heat shock protein 60 is in the mitochondrial matrix here. So all that can be quite easily now resolved. And here's an example from the Golgi complex. We nicely see here a protein on the rim of the Golgi. And just 2 more minutes, if I may. So we can also use this in tissue. This is a brain tissue section, a mouse brain tissue section. And if you zoom in here, you can easily resolve the synapses here, postsynaptic density, and the dense projection on the presynaptic side here at these locations. And this is published in a bioarchive paper with many, many different examples if you're interested. And then the last thing I want to show is here this 3D stack of this proximal tubule in the mouse kidney. This was done by Ons Amsad, the student who invented that in my lab and who is now running the company here. And this was actually done in her new capacity, but she let me allow her to use this. So we can now scroll through 3D here now through that proximal tubule, and we are reaching here the brush border in a second. And I think you here, I think it's quite a beautiful kind of 3D structure that becomes nicely resolvable now just with your confocal microscope, right? This is not electron microscopy. This is just imaged on the confocal instrument. You saw here the mitochondria with all the cristae here everywhere. I think now we're going through here these microvilli here. Okay, that's just here a summary of the expansion microscopy, and then I promise I'm at the end. So the advantage is it does not require any specialized microscope. Any confocal microscope is sufficient. Works with cells and tissue, 3D imaging very easily. And if you use the pan expansion microscopy version, you see that ultrastructural context similar to the electron microscope, and you get correlative light electron microscopy-like images. Disadvantage, the sample preparation process is fairly involved. It takes several days to prepare a sample, and you need to validate that what you see is actually expanded correctly. So especially if you work with new tissue, new protocol, you need to go through that step. And of course, it's not lifestyle compatible. That's it. I apologize for going over, but I would be happy to take any questions you might have.
Thank you, Joerg. That was a great talk. Anybody have questions that they want to unmute themselves and ask?
I do.
Ken, go for it.
So I think, Joerg, if you had started with the padluminant slides first, you would have lost your audience by the time you got to the rest of the super resolution techniques. I kind of don't see. I mean, it just seems like this seems like the future. Is there some issue with the labeling? I mean, what you mentioned some of the downsides. How you said something about the post-expansion labeling. Is that challenging, or is that not?
It's not trivial, but so not every antibody works because we denature our proteins. So about half the antibodies that we try work for us. And often it's actually the antibodies that work well in Western blot, right? That work for us here.
Makes sense. Yeah.
It
But it's
doesn't-
nice that you can actually identify the antibodies you want to use that way, right?
Yeah, so we haven't done it systematically, but that's something we still need to do. So yeah, that's one of the problems. The other thing is you need to optimize it really for each kind of tissue. So we have figured it out for cultured mammalian cells. I think that works really well. We are now working with Diane Krause on working also with non-adherent cells, like here blood cells. So I think we are getting there. The brain tissue needed some. You always need some adaptation, and then if you work in smaller model organisms like flies or worms or so, then it becomes harder because they are built to resist kind of harsh chemical environments, right? So that requires quite some sample optimization, and then the process is slow, right? So the full pan expansion microscopy protocol, including antibody staining, can take a week.
So one other question, I mean, and I'll get out of your way. So in the context of tissues, do you have any way of fusing different scales of imaging? In other words, it would be a lot to image a glomerulus, a kidney tubule, any large structure at that kind of resolution. But do you have an idea in mind about how you would perhaps fuse data collected at two different spatial scales and resolutions such that they could be in the same space?